Hello there, welcome to this first lesson on prokaryotes and eukaryotes for Access to HE, Human Physiology and Introduction to Cell Biology. This is one of your first criteria on your assignment, and you're asked to compare the ultrastructure of a prokaryotic and eukaryotic cell. So we'll be going through what each are, the common features of them, and we'll be looking at the end to see a comparison. And when we're looking at a comparison, we are looking at the similarities and differences between the two. For a long time now, scientists have argued about life and about the characteristics of life, but they've generally agreed on 6 or 7 different characteristics that all living things share. If you were to compare something inanimate like a chair or a table and compare it to something that's living like a human or a dog or a tree, then you're going to see set differences, and those differences we've talked about in class, or should have talked about in class, where you would see that living things respond to stimulus, whereas plants and trees respond to light. We respond to a T-Rex charging at us, for example. All living things grow, they evolve, they have some kind of metabolism, and importantly for us, particularly in terms of your studies for the rest of the year in human physiology, all living things are made of cells. Now, this first slide really is important because what I wanted to explain to you is why this criteria is like it is for this first part of the assignment. Compare the ultrastructure of a prokaryote and a eukaryote. So why has the awarding body chosen that as a starter for your studies? And this table down the right might give you some kind of explanation. This is from Cavalier-Smith, who's a famous scientist who came up with a classification of living organisms, and you can see that this is the 6 kingdom classification. But the first differentiation that he makes is that living things are separated into prokaryotes or eukaryotes, and that's why this sort of study, this topic, is very important, because it's the first distinction of living things. You're either a prokaryote or a eukaryote according to this system. I'm saying that because the system's likely to change, and like I say, there is an earlier model, I think something around about 2005, where the same person, Cavalier-Smith, come up with a 7 kingdom classification. I'll explain that in a second. But I thought it was really interesting and important for you to understand why this is a topic, because of all living things, the first distinction is made between whether you're a prokaryote or a eukaryote. Now, according to the table, prokaryotes have 1 kingdom, which is bacteria. This has changed to in the 7 kingdom, and actually archaea is on its own. It's a separate prokaryote, and so this gives rise to the 1, 2, 3, 4, 5, 6, 7 kingdom classification. All right, so prokaryotes, according to this classification, are either bacteria or they're archaea. Under the eukaryotes, you then have fungi, animals, protista, plants, and chromista. All right. Obviously, for our studies for the full year, we will be talking about animal cells in the main. We will be talking about prokaryotes and bacteria in our infectious disease unit, but mainly we'll be talking about these cells. So the reason why you're studying this early on is so that you get a grasp of what is the fundamental difference between a prokaryote and a eukaryote, and what are the similarities and differences between them. Interesting to note that a virus is not on the list. It's not in the classification. Technically, it's not alive. It doesn't share many of the characteristics of life that we talked about in class. It does have some, or it behaves as though it's alive, but some of those classifications are not there, so it doesn't form part of this classification of living organisms. So before I tell you, or before I explain to you the similarities and differences of a prokaryote and a eukaryote, it's really important that you know the name and the function of each of these different features of a cell. If you don't understand what each of these things do and their names, then there's a good chance that you're not going to fully understand the difference and the similarities between a prokaryote and a eukaryote. So my intention with this video is that I'm going to take each one at a time, break them down very simply and explain what they do and their differences between prokaryotes and eukaryotes. And then at the end I will summarize and tell you a little bit about what the research is saying. Okay. If you already know these features of cells and what they do, you by all means skip to the end to see the similarities and differences, and you can move on quicker. Okay, the cell membrane and cytoplasm, and indeed cytosol. Cell membranes are made of phospholipids, which we'll do in class, and the cell membrane is the thing around the outside. It is not the cell wall. Cell wall is very different. So here we have a eukaryote on the left and a prokaryote on the right. So eukaryote and prokaryote. Now notice that both types of cell have a cell membrane, and that's one of the similarities between the two. Structurally, they're slightly different. So structurally, they're mainly the same, but slightly different. And I will leave that to your own research if you can find out. So this is the cell membrane, and this is the cell membrane. They're made fundamentally of the same stuff, but they have slightly different ingredients in them. The thing that fills the cell is fluid, and it's generally the same kind of fluid, and that is the cytosol. Mainly water, but it has other things in it like amino acids and sugars and stuff like that. And then what's referred to as the cytoplasm is the cytosol with different things in it, like membrane-bound organelles in it. Okay, so that's just an important difference that you might see in a cell. That's a cytoplasm. Okay. An interesting difference between the eukaryote and prokaryote is that sometimes the prokaryote might have a capsule around it, sometimes called the slime layer. Be careful when I'm drawing it, but yeah. So there's a cell membrane with a slime layer around it. Now we'll do lots of work in infectious disease about what that slime layer is and does. But that's a difference between prokaryotes and eukaryotes. And sometimes as well, that a prokaryote might have another cell membrane around that. So it might be like a slime layer sandwich around the outside. And generally you won't find that in eukaryotes. So that's a fundamental difference. And to sum up this slide, they have them, but they're slightly different in composition. But also the prokaryotes can have an extra couple of layers around it, the slime layer and the extra cell membrane. So this slide is about the genetic material, and the genetic material usually is DNA or RNA. Hopefully you've heard of that before. We're doing a lot more of that in the macromolecules unit. But the genetic material is one of the most important differentiations between a eukaryote and a prokaryote. So the DNA in an animal cell or a eukaryotic cell is generally linear, and what we mean by that is you'll have seen this in movies, science fiction movies. It has that double helix, that classic double helix shape, but it's in a line. All right, that double helix can go on for a very long time, and we call that a chromosome. So one of those lines is a chromosome. Now, in a eukaryotic cell such as a human, we have 23 pairs of these chromosomes, and they're huge. So we have 46 chromosomes, and they're all vital. They all contain the code to make that organism. So your chromosomes in any one of your cells contains the information to make the whole of your body. So if I pull out a cell from your big toe, for example, lovely image, we can extract the DNA, and all in that DNA gives all the instructions to make your whole body. Okay, all of the DNA is the same in every single one of your cells. Now that is vital information. We can't have those chromosomes wandering around freely, otherwise we'd get severe genetic defects and we wouldn't be able to replicate ourselves. So one of the first evolutionary changes that we saw is that in a eukaryotic cell, a membrane was put around DNA to keep it together like a vault. Okay, so that's a nuclear membrane. Nuclear membrane. And that is the nucleus. The nucleus. So the whole thing is the nucleus. The thing around it is the nuclear membrane. Sorry, I'm writing with a mouse. Okay. Now, in a prokaryotic cell, the genetic material generally tends to be not linear, it tends to be circular. Usually one circle, but they can be 2 or 3 from what I've read. But they tend to be a lot smaller as well. If you can imagine that prokaryotes are 1 cell big, this is as big as they're going to get, 1 cell, whereas eukaryotes such as humans are trillions of cells big. So the information that the genes have, the information on that DNA and RNA is massive in comparison to a prokaryote. So generally the DNA in a eukaryote is there are more, it's bigger, hence the linear formation in terms of a double helix strand rather than a circular bit of DNA, which is the same structure. It's still double helix, but it's just in 1 circle. And notice that the circular DNA in a prokaryote does not have a nuclear membrane around it. It's free. So that is one of the biggest distinctions between a prokaryote and eukaryote. Does it have a nuclear membrane around its genetic material? If it has a nucleus, it's going to be a eukaryote. If it doesn't have a nucleus, it's going to be a prokaryote. The other thing to mention is that some prokaryotes have like small rings of DNA, really miniature rings of DNA, and they're called plasmids. And they're also, they're generally not in, well, they're not in eukaryotes, they're in prokaryotes, and they have a particular reason, but that's also possibly RNA or possibly DNA. It's genetic material, but it's not the main chromosome. It's not like the main genetic material. This big ring here is the main genetic material. Prokaryotes have some miniature forms of DNA which can pass on to other prokaryotes and things like that. So to summarize, the genetic material is generally the same, DNA or RNA, but in a eukaryote it's locked up in a nuclear membrane in a nucleus, whereas a prokaryote is not. The genetic material generally tends to be bigger in eukaryotes and smaller in prokaryotes, and prokaryotes can have genetic material in the form of plasmids, whereas eukaryotes don't. So on this slide I want to talk about free ribosomes, and both prokaryotes and eukaryotes have free ribosomes. So I'm only going to draw a eukaryotic cell here, and I'm going to draw these free ribosomes here floating around the cell. They're called free ribosomes or free floating ribosomes because they can just float around the cytosol. They can go wherever they want within the cell. Okay, they're not attached to anything. And ribosomes in general are sites of protein synthesis. Protein synthesis, which simply put means that they are the protein factories. This is where proteins are made. Now, I don't think, or it's possible that you don't have a full-on grasp of how important and what proteins do for our bodies. If you think about every neurotransmitter that you can think of, dopamine, adrenaline, acetylcholine, they're all proteins. Think about all the hormones of our bodies, estrogen, progesterone, testosterone, prolactin, anything like that. It's a protein. And without proteins, a cell becomes pretty useless, and we need to be able to make them to our own specification in order to grow. So free ribosomes create proteins for the cell to use. Cell use. So free ribosomes create proteins for the cell to use. So if this bottom left ribosome here was creating a protein, here's the protein, then the cell would use it. It can't get out of the cell, and this protein cannot be used by the rest of the body. It will not be used because it's been made at a free ribosome. And there is a clear distinction, which I'll explain later on. Now, why would the cell want to create a protein, and what example can I give? Well, let's have a look at a protein channel. Here is, in green at the top there, a protein channel. That's just one protein. I know there's a gap in it, but it is one protein. And a protein channel allows a molecule to pass through the cell membrane that wouldn't usually be allowed to get through. So if this molecule here, this square red thing, bumped into the cell membrane, it wouldn't be allowed to pass, and therefore it would move elsewhere. But if it came up with this protein receptor, if I can zoom in, here's a protein receptor looking down the sort of barrel of the receptor. In a very, very simple term, it's like a kiddie's toy. Oh, I wanted the red. Here, that's the hole in the protein receptor. So anything without that shape, let's say it's a moon shape, like one of those kiddies' toys. If that moon shape was to try and go through this tunnel, it wouldn't fit. But because it's got the right corresponding shape, this red square, should do it really in 3D, then that's going to be able to fit through and get into the cell. Now it's exactly the same of what happens here with this diagram. This square fits that shape and it is allowed into the cell. All right. Now here's another shape, but it won't fit through the cell because it's not, or it won't fit through the channel because it's not the right shape. So that's what I mean by a free protein making a, free ribosome making a protein for the cell itself. And a protein channel is an example. So how on earth does the cell make such a shape that's corresponding to the right shape of the molecule that wants to get through it? Well, the clue is in the DNA. So here we have the nucleus, and here we have the DNA of a cell. Now the DNA of a cell contains the information to make all of the proteins in your body. So let's say this section here is the code for that protein. So this section, this code here, codes to make that shape of a protein, which is required over here. We want to put it on this space where I've left on the cell membrane. Now the DNA is locked inside the vault. It can't go to the site of protein synthesis. It can't go to the ribosomes down here because it's locked in this nuclear membrane. So what it has to do, it copies this code right here. This yellow line that I'm making here is the copy of the code. It's like the recipe. It's like going to a library and photocopying a recipe of a cake that you want to make. So that recipe needs to go to the kitchen, and this recipe can leave the cell and go to the ribosome for the cake to be made. This is where the ribosome comes in, because what happens is the ribosome grabs the ingredients that are floating around the cytosol. It goes, oh, I need you, 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 in the right order, because the right order is from the DNA. The DNA doesn't change. So it's like making like a Lego structure. If you follow the instructions, you're going to make the same protein every time. And what happens is this ribosome eventually produces the protein channel that we wanted up here. Exactly the same protein channel. Which I find absolutely incredible. It's going on in your cells right now, all day, every day, putting receptors on there, making enzymes, making all these different hormones, all these different neurotransmitters. But the problem is, how does this protein receptor get to where it wants to go? It wants to go up here. Well, I'll tell you later on another slide in more detail, but basically another protein comes along and basically drags it into position. So now we have a receptor protein on the cell membrane that we wanted, which will then allow these red squares to get in. And that is what a free ribosome does. It provides it, or it produces protein for the cell itself, not for export. It's quite an unusual choice to talk about this next common feature of a cell, but we're going to talk about vesicles, and vesicles are basically transport bubbles. Transport bubbles. So if we drew a eukaryotic cell membrane, we would draw vesicles as sort of relatively spacious and large bubbles. Okay. Now they perform a really important function in that the vesicles are made of the same stuff as the cell membrane. So you may well have heard, or may well not have heard, that this is a phospholipid, all right, and the cell membrane and the vesicles are made of phospholipids. So the cell membrane, or the plasma membrane, is a double layer. So that, you know, that is the phospholipid bilayer, and that is what makes up the cell membrane. And the vesicle is made up of a single layer of phospholipids. So these sort of heads just form a circle all the way around to form a transport bubble. That is what makes up the vesicles. Now, it's really important because anything a vesicle can get out of the cell. So remember our ribosomes from the last slide. Here's a ribosome, and if it created a protein, let's say it created this protein here, and if this protein was dragged to the cell membrane, or it moved to the cell membrane, because it's large and insoluble, it wouldn't be able to exit the cell. It wouldn't be able to exit the cell. But if we put a vesicle around it, that's a large vesicle, but it's a vesicle nonetheless, then it can exit the cell. And if you've seen the video on active and passive transport, you will know how. Let me draw the membrane here. So if this vesicle comes towards the cell membrane, it can actually bind to the cell membrane because it's made of the same stuff. It just merges in, and that membrane then can open up, rip apart, and that molecule then can exit the cell. So vesicles are really important in transporting things into and out of the cell. It works the opposite way. So this is exocytosis, and the alternative view would be, let's say, a molecule here pushing up on the cell membrane. The cell membrane would sag around it, and then it would pop off it into its own vesicle. So vesicles are really, really important for moving stuff inside and out of the cell. Now this is called endocytosis because it's going in, and exo going out. So that's the importance of vesicles. And I wanted to show you that really for the next slide, setting you up for the next slide, because we've talked about free ribosomes, and free ribosomes produce proteins for that particular cell. But the question is, what if you're a gland? What if you're the adrenal gland, which produces the hormone adrenaline? How is that going to escape the cells into the bloodstream, travel around the bloodstream, and engage your muscles? So there has to be some way in which proteins can be made by a cell to be exported out. And I'm hoping this gives you a big clue as to how proteins are exported out of the cell. Well, we package them in vesicles, in transport bubbles. And so you'll find that certain cells are experts in packaging proteins in these vesicles, so that at any point in time, thousands and millions of these vesicles can fuse with the cell membrane and spill out its contents into the bloodstream. So let's have a look how that happens. Okay, so building on what we've just said then, the rough endoplasmic reticulum, or the RER for short, and the Golgi bodies are basically the method by which hormones or proteins are exported out of the cell. So let's have a look. This is our nucleus in the bottom corner here, and here is a folded stack of membranes, very, very close to the nucleus, and on that membrane are a series of ribosomes, which is why it's called the rough endoplasmic reticulum, because it looks rough because there are things studded on it. Okay, there are these ribosomes, when really, in a common sense term, the rough endoplasmic reticulum is a membrane with ribosomes on it. Remember, these purple things are ribosomes. So the rough endoplasmic reticulum, in common sense terms, is a membrane with ribosomes on it. Okay, so we're then going to draw these things here, which are the Golgi bodies, and then around the outside of the cell I'm going to draw the cell membrane. This is the plasma membrane. Now remember, I just want to do a recap. The free floating ribosomes are here. So what happens is we take a copy of the DNA, we take it to the ribosome then takes the ingredients, chains them together to make a particular protein, which then can go onto the cell membrane. All right. Now, it's essentially the same way, but what we need to do by the end of it is package this protein in a vesicle, and that is what the job of the Golgi bodies is to do. So, if a cell is going to package a protein into a vesicle, the same process occurs. We take the copy of the genes, of the code from the DNA, because remember, the DNA is too big, too important, it can't leave the vault. The copy then goes to the ribosomes. Now the copy is mRNA if you're interested, but don't worry if that doesn't make any sense. And the ribosome is really not free floating around, it's studded on the membrane. So it's not going to go anywhere, but it does the same job as this thing over here. it pieces together the amino acids or the building blocks of protein in the right order and puts them together on the inside of this membranous sac. So it's not in the cytosol. This protein has not been made in the fluid in the cytosol of the cell. It's been made inside that rough endoplasmic reticulum, inside this membranous sac, the rough endoplasmic reticulum. So I'm going to give this this green protein here, I'm going to give it the H for hormone. Okay, so this here is the H. This is a protein. It's made of amino acids, and that hormone will travel through the rough endoplasmic reticulum, and it'll go through into the Golgi bodies. Now the Golgi bodies, exactly the same, are made of phospholipids. They're made of the same stuff of the cell membrane. So what happens is this hormone pushes out of the membrane, into its own vesicle. So basically, if this was the adrenal gland, a cell of the adrenal gland, we take the copy from the nucleus of the DNA, we take it via mRNA to the ribosomes. The ribosomes grab the ingredients from the cytosol and push this hormone into the endoplasmic reticulum, the rough endoplasmic reticulum. It travels through the rough endoplasmic reticulum to the Golgi bodies and is packaged into a vesicle. When this vesicle then gets dragged to the cell membrane, when it's triggered to, when we have a fight or flight response, for example, then it triggers exocytosis of this hormone. Now there's going to be thousands and millions of these vesicles. I'm just drawing one, but essentially the vesicle opens up, fuses with the cell membrane, and is then free to leave the cell into the bloodstream, and that'll travel around and bind with receptors into your muscles. And that's how proteins are packaged. Just as a side note, sometimes there are proteins that are put on the cell membrane here, let's say. Now if you have done the work on the cell membrane, we also have proteins in there that might have sort of, let me draw them in orange, we might have sugars coming out of it, we might have fats coming out of it. Okay, so a cell, if you were to look at a cell down here on the right hand side, if that's a cell, they have kind of things poking out, receptors poking out of them. If you looked at a cell membrane on the outside, you know, and that's called its glycocalyx, it's called its sugar coat. And if another cell was to come along and bump into it, if they bump into it and recognize it, that's how cells communicate. Well, why am I telling you this? Is because the Golgi bodies are responsible for adding these things on. So the protein comes through the rough endoplasmic reticulum, gets to the Golgi bodies, and the Golgi bodies modify the protein. So let's say this red dot here in the Golgi bodies is a protein. It's just been made by a ribosome on the rough endoplasmic reticulum. It travels through the network of membranes into the Golgi bodies, and the Golgi bodies has been informed that it needs to add on some sugars to it, and that's what it means by modifying proteins. And all that would happen would be that would come out in a vesicle. Let's draw the red and the two greens. This vesicle then would fuse with the cell membrane, just like we've seen, but rather than escaping to the bloodstream, this protein here with its sugars poking out will then present to the outside world and tell the cell, communicate with other cells around it. And that's the rough endoplasmic reticulum and the Golgi bodies. Okay, so let's talk a little bit about the smooth endoplasmic reticulum. The smooth endoplasmic reticulum looks very much like the rough endoplasmic reticulum. However, there is nothing studded on it. There are no ribosomes on it, which gives you an impression that it must not be for protein production. If there's no ribosomes, there's no protein production. Well, what does it do? It basically stores and produces lipids. So that could be phospholipids, for example. So we know that all the organelles in a human cell, like the rough endoplasmic reticulum or the lysosome or the mitochondria, have membrane around it, which is made of phospholipids. So this thing stores phospholipids, head and two tails. And that can be used in the cell membrane itself to create new membrane. It can be used to create membrane around another organelle. It has also another function of metabolizing harmful products. I think, if I'm right on the top of my head, it also helps in the neutralization of alcohol. But for us, I'm going to say the main function would be to store and produce lipids. What I will say to you is at the end of this then is that the organelles that I've talked about, i.e. the Golgi bodies, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, all have membrane around the vesicle. Sorry, not membrane around the vesicle, membrane around the organelle. And this is something that the prokaryotes don't have. They don't have mini organs inside the cells surrounded by vesicles. Sorry, surrounded by cell membranes. Dear me. All right, so that's a key difference between prokaryotes and eukaryotes. So let's talk about the lysosomes, and another membrane-bound organelle of the eukaryotes. Essentially, they are the police of the cell. Whoop, whoop. So, a lysosome will have digestive enzymes inside it. So, if there's an organelle like a mitochondria, for example, that's misbehaving, it's at the end of its life, it's not doing as it should, this lysosome then will come over, fuse with it, and insert its digestive enzymes. And this will start breaking all the bonds down in the atoms, and it will start recycling. It will start breaking down the proteins into amino acids, etc. And those amino acids will float around the cell, ready to be used again and reassimilated by the ribosomes. So nothing goes to waste. You know, the ribosomes, when they're making the proteins, will go, oh, there's an amino acid, I need that one, bring it in and start making a new protein and building another organelle, for example. So very useful are the lysosomes. Okay, the mitochondria really is specific to animal cells, not really particular plant cells. They have chloroplasts, and certainly not prokaryotes. Here is the cell membrane of an animal cell membrane. Here's the nucleus. Now a mitochondria looks sort of like a bean shape, and you'll see that it has a squiggly line in the middle and a sort of internal compartment. That will be the mitochondria, and they are involved in energy production. And they actually sites of ATP, it's called, which is our body's currency of energy, adenosine triphosphate. So they recycle our ATP stores to give us energy, and that's really it, to be honest. They're membrane bound, so that's why they're not found in prokaryotes. They have a phospholipid envelope around them, and what happens is when you breathe in oxygen, the oxygen travels to each of the cells around the bloodstream, along with the glucose to your mitochondria, and we use that oxygen and glucose to turn these things into ATP. The ATP is what the cell can use to start pumping stuff out. It can use it for the exocytosis, which we've seen, or endocytosis, which we've seen. It's the ATP that is the energy currency, so that's what we can use for energy. The cell can't use oxygen or glucose directly for energy, so it can't just take oxygen over here and just use it to create energy. It has to go to the mitochondria to be burned, essentially, to create ATP, and that's what it is. Okay, this feature of a cell often gets overlooked. The cytoskeleton and microtubules. Well, the cytoskeleton is made of microtubules, and really the clue is in the name. It's a skeleton of the cell. It's the bones of the cell almost. So if we drew a cell like this, then all the way throughout it there are proteins that span the network and the cytosol, which keep it in sort of a generic, in a sort of agreed structure, and they'll pretty much mostly lead to the nucleus, so that when things well, first of all, to keep its shape. So if we look at, I don't know, let me just choose one, a sperm cell, for example, it's got a real specific shape. If we look at a nerve cell, then it's got a specific shape. And we can choose any cell that we like, and these cytoskeletons give, like scaffolding poles that give the cell its shape, and they're made of proteins. So these proteins are made at the free ribosomes, and they just help keep the shape of the cell. Now, it's not just about shape, it's also, this is incredible, and you can look it up on YouTube, and there are videos of this slowing down. We have other proteins, motor proteins, that basically walk up and down these like highways. I'll try and get a video in the PowerPoint for you, but they use them to sort of tow things around. So if there's a vesicle here, it latches onto the vesicle. So it's a hormone, and it actually takes it to the cell membrane, which is absolutely mind-blowing. We've got stuff walking around ourselves like a science fiction movie, and I recommend the BBC documentary that I've put up on the Google Classroom, The Hidden Life Inside a Cell, and it will show you these things in action. It's incredible. But the cytoskeleton is there for like highways, motorways, for things to get around the cell, and it's made of microtubules, which are proteins that are made at the ribosomes. They're made of amino acids, just like everything else, and which is why it was very important for me to establish the role of proteins and protein production. Okay. Interestingly enough, that both prokaryotes and eukaryotes do have cytoskeleton. Again, they're slightly different in terms of the composition of the proteins, but you can write about that in your assignment. Okay, so to explain these properly fully, I think it would be useful, once you've had a look at this, to have a look at the mitosis and the meiosis videos to really get a flavour of what they do. But if I draw there's a cell membrane, that's actually a cell membrane, and I draw the nucleus, so the DNA is inside. Centrioles, well, there's one centriole, and centrioles are vital for cell replication and mitosis. So what happens when a cell is going to go through cell division, i.e. it'll grow or replicate, then we get two centrioles, so they replicate, and two of the centrioles make a centrosome. Very confusing, two C words, that's often how it is in human phys. And what happens is these two centrioles move down opposite sides, and then they start ripping things apart. And go into the mitosis video if you want to find out exactly what happens. But centrioles and centrosomes are very much involved in mitosis, in cell replication. They're also involved in moving things around as well in terms of the cell, and very much involved with the Golgi bodies. But this is a very basic and introductory video, so yeah, if you want to divert your own research, that's fine. But you will only find these in eukaryotes, because eukaryotes replicate through mitosis. Prokaryotes mainly replicate through binary fission, which is a completely different but similar process. Okay, so these three things are quite familiar across prokaryotes and eukaryotes, so they do get crossed over. There are similarities, but when I sort of see learners using these terms, they get quite confused. So let me draw a prokaryote. Typical bacterium. Oh, when I say typical, it's a stereotypical one that you see. Now, a flagellum is this. It's like a tail that whips from side to side. That's the flagellum. Okay, now where have we seen that in a eukaryote? Well, I've just drawn it on the other slide in the sperm cell. Okay, so we see it in prokaryotes and eukaryotes. Cilia or ciliated cells are like fine hairs on the side of a cell. Cilia. Now, in prokaryotes, cilia are used to sort of attach to host cells when they're in the body or when they infect. They can also be used for a little bit of movement, but generally they just beat one way. So if something, I don't know, if something was grabbed here, this thing would move forward, move forward. So you'd end up being able to move, and that can actually move something, or it can move itself. In terms of humans, there are cilia on the fallopian tube of women. So when the egg is released, cilia beat on the fallopian tube, which enticed the female egg down the fallopian tube. Also, if this was your throat, and sorry, your throat, that's a shocking scientific term. If this was your trachea or your esophagus, let's go for your trachea, and you were breathing down this tube, you'd have cilia down lining this. You know, you'd have cells that line the airways like this with cilia on it, and that's in case you get anything down that tube, or you get mucus or anything like that. It traps the particles of dust and air, and these cilia beat up towards the back of the throat so that you can cough it out, basically. So cilia are definitely found in prokaryotes and eukaryotes. Microvilli are even smaller. They're super small, and microvilli are therefore an increased surface area. So the best way I can put it is, if you've got a cell like this, I'm going to draw 3 cells. And if we measured this distance here, let's say it's 3 centimeters, then what we can do is we can put folds in it like this. And if we measured up, down, 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 we might have increased it to 7 centimeters, let's say. Now, microvilli, you'll often find cells that have folds in the membrane like that, but then have microvilli on top. So each one of these folds, you have minute microvilli pointing out all the way along. Forgive the crudity, but if you measure this microvilli up and down, inside, just like in and out of the different gaps between the microvilli, you may well see that it's something like 20 centimeters long. And so from the same width of space, you get much more surface area, and that helps things to go in and out of the cell, for example, increases uptake. So This is definitely in eukaryotes, and I'm pretty sure, in fact I'm 99% sure you do see that on prokaryotes too. So microvilli, flagellum and cilia are quite similar in structure because they're sort of appendages out of a cell. They stick out of the cell, but they're different in structure and they do different jobs. The flagellum generally for movement, this whip-like tail. The cilia is for movement, but they don't move massive amounts. They beat rhythmically to move things along, and the microvilli are there for increased surface area and exchange of material. Okay, so if hyperlink to the Khan Academy there, the Khan Academy has been around for a while, and I recommend them for learning resources. They're superb, very much in a similar sense to what I do or try to do on the PowerPoints. But if you want another view, if you want a more Americanized view, if I say, then check it out. It's all good. Okay, so the criteria of the assignment of the unit is asking you to compare the ultrastructural features of a prokaryote and eukaryote. So it's very important that you're not just looking at the similarities, you're also looking at the differences too. Now a table like this from a really reliable textbook is perfect because it shows that you've researched the information. It's too easy for you to do a quick Google search and go to, for example, Biology Mad, Life Science, etc., etc. If you use those sources in your assignment, you won't fail, but get into good habits now and look for a published, reliable source. Now, I'm not saying that all sources are unreliable that are on the web, but we want as high a standard as possible. So this is out of a textbook, and it summarizes the similarities and differences. Now, you could use a table like this, but what would be expected is that you write about what the table shows. Now, the other thing that I would suggest is that you don't write anything that you don't understand. So for this introductory unit, I would not expect really that you would be talking about transcription and RNA polymerase, for example. Now, I'm not suggesting that you don't understand it, but it's something we're not expected to be covered at this point in time. And therefore, if you write that in your assignment, there's a good chance that you're not going to show that you understand it, and it's all about your understanding. So pick out the similarities and differences using a table such as this, and the more reliable sources, the better quality assignment that you're going to put. So let's just go through it together. We can see that the prokaryotes are here, 1 and 2, and the eukaryotes are in this final column. So typical size, that you can see that the prokaryotes are small, the eukaryotes are big. So that's a difference. Nucleus and organelles. You can see that the prokaryotes have no nucleus and no organelles, but the eukaryotes do. So that's a difference. The microtubules and filaments, which make up the skeleton, you can see that it's actin-like, and tubulin-like. They're types of proteins. Okay, and then these are made of actin and tubulin proteins. So it's also a similarity and a difference, in my opinion. They both have microtubules and microfilaments, but they're kind of made of different things. Exocytosis and endocytosis. I would expect you to understand what those two words mean. I have explained them briefly in this video, but if you go to the active transport video session, it will explain it in loads of detail. And you can see no, no, and yes, it's a difference. So the eukaryotes are able to exocytose and endocytose material, whereas the archaean bacteria or the prokaryotes cannot. That boils down to the vesicles, you know, and the Golgi bodies. The Golgi bodies are able to produce vesicles, whereas the prokaryotes don't. Cell wall. It's possible for you to be able to explain this in your assignment, but you don't feel like you have to. You could look into what peptidoglycan means. Now, this is comparing it really into plants and fungi. There is none in human, so we don't really have a cell wall in a human. So this is human physiology. It's not particularly necessary, but it is a comparison to make between eukaryotes and prokaryotes. The mode of cell division, I did talk about mitosis. Well, here we are, mitosis. That's how we go through cell growth, and we go through meiosis for production of sperm and egg or gametes. Have a look at my videos on mitosis and meiosis. That will give you a real in-depth knowledge of how cells grow or how we produce sperm and egg. But you can see that prokaryotes go through binary fission. So that's a very much difference between prokaryotes and eukaryotes. Find out what binary fission is. Have a research about it. We'll definitely be covering it in infectious disease, and if you look at before we get to infectious disease, that's going to help you along the way and have less learning. The typical form of chromosomal DNA, you can see that it's circular and for few associated proteins, circular with histone-like proteins, and linear, which we talked about, chromosomes. Circular, circular, and linear. That's a difference we've got there. Let's not really talk about RNAs, etc. We've got the ribosomes, which we talked about. It's a similarity and a difference. Explain it. They do have them, but you can see this is 70S with 55 proteins, this is 70S with 65 proteins, and this is 80S with 78 proteins. And all that means is that they're bigger in eukaryotes. The actual ribosome, which I drew like that, whereas that might be this size. I mean, that's not a realistic size. They're like in a V shape, really, I think. But I'm simplifying everything in this video. If that's a ribosome in a eukaryote, that might be the size slightly smaller in archaea, and that might be the size in a bacteria. So you can see that's what we mean by 55 proteins, 65 proteins, and 78 proteins in just different size. And the 70, 70, and 80 is a subunit. So by all means, have a look. It's understandable, but it's not quite necessary for us at this time. Now, it's really interesting that I did some extra research on the difference between prokaryotic and eukaryotic cells, and I came up with this journal article here, which was from 1999, I think it was, which is quite, it's quite a long time ago, but it came up with some very, very interesting things. So the title was "The Origin of Eukaryotes, the Difference Between Prokaryotic and Eukaryotic Cells," and the references down here if you want to check out the article yourself. Interestingly enough, that this is a really nice quote to start with. "The difference between a prokaryotic and eukaryotic cell structure is obvious. Fundamentally, all eukaryotic cells have a membrane-bound true nucleus, a complex endomembrane system, and cytoskeleton, whereas prokaryotes do not." That's an interesting quote there, saying that all eukaryotic cells have a membrane-bound nucleus, which we said, a complex endomembrane system. That's the things that we talked about, the internal organelles, so the mitochondria, the lysosome, smooth endoplasmic reticulum, rough endoplasmic reticulum, mitochondria, etc. They've all got membranes around them, whereas prokaryotes don't. And a cytoskeleton, which is contradictory to the table that we just looked at. So there's some kind of ambiguity here in terms of science and where things fit in, and where, if we found a prokaryote with a nucleus, is that going to smash the whole classification out of the water? But if you read further on into this, some bacteria have been reported to possess a nucleus, a cytoskeleton-like structure, and perhaps mitotic-like apparatus, a complex internal membrane system, a linear chromosome, introns, a eukaryotic-like gene regulation mechanism, or multiple genomes. So actually, you read on in that article, and it's contradicting the classification system a little bit there. It's saying that some prokaryotes have been found to have these. So I would say, even though it's a scientific discipline, science can be disproved every now and again if things crop up that you're not expected to see. And science is based on everything that we know at this point. And I like to pick on this last paragraph here that's at the bottom. There are at least 2 cytological features that have not yet been found in bacteria. They lack any energy-producing organelle, and they are not capable of endocytosis, which we've talked about. So I just thought I'd throw this in at the end there to try and open your minds, really. And sometimes the textbooks can be out of date, or they don't present the whole picture. And it's down really to us and to you independently to read into the subject and find out what you can, because sometimes it's not as crystal clear as you might think.